Hello everyone, Mr. Linder here. Let's talk about steroid hormones and their mechanisms of action. And then let's also look at hormones that are similar in action to steroid hormones, but aren't actually steroids. So first off, let's talk about steroid hormones. So steroid hormones have similar structure and are based off of a model of cholesterol. So cholesterol is actually the precursor to steroid hormones. So if we take a look at this diagram, you'll notice that this is the precursor cholesterol molecule. And if you use enzymes, so this diagram has several enzymes listed, aromatase, 21 hydroxylase. Of course, enzymes are used to catalyze each reaction in this pathway. And some of the molecules you notice aren't even mentioned in this diagram because this is not an organic chemistry class. But you can take cholesterol and you can produce these various precursors using enzymatic reactions. So we could just write E here for enzyme, and I can put E here for another enzyme. And what you're seeing is cholesterol can be converted into steroids like progesterone, and then we can make other precursors and then produce corticosterone, et cetera, et cetera. We can make things like aldosterone. But again, remember, each reaction has an enzyme that's going to catalyze it, but we're all starting with the precursor cholesterol. So when you're making testosterone, or you're making estradiol, or you're making cortisol, aldosterone, these are all steroid hormones, and we utilize enzymes to generate these steroid hormones from the precursor cholesterol. Now, there's only a few organs that are actually going to produce steroid hormones. So we have the adrenal glands that produce steroid hormones, and specifically that would be the adrenal cortex. The medulla, the inner portion of the adrenal gland, will make things like epinephrine and norepinephrine. But it's the adrenal cortex that's going to produce your steroid hormones. Gonadal tissue, so ovaries, testes, these are going to produce your male and female sex steroids. And then you have the placenta, which is only produced during pregnancy, and that too is going to produce steroid-based hormones. So there's only a few glands that actually produce steroid hormones. When you look at synthesis of steroid hormones, you're going to notice that the cells that synthesize steroid hormones are going to have large amounts of smooth endoplasmic reticulum. And so the smooth ER within the cells, so we could actually, if we had a cell that was actually synthesizing, let's say that you had a cell in your adrenal cortex, there would be a lot of smooth ER in that particular cell, and it's going to utilize the cholesterol and it's going to utilize enzymes. So we're going to take cholesterol and we're going to use enzymes to produce these steroid hormones. And then those steroid hormones can be released into the bloodstream. So we have in this diagram, we have a steroid hormone in yellow. It's circulating in the bloodstream. But because they are lipophilic in nature, meaning that steroids are fat-loving, they're not going to circulate in the blood without assistance. So because of the lipophilic nature of steroids, you're going to produce them on demand. You're going to then release them into the bloodstream, and then you're going to need a carrier protein in the blood to move those steroids around. Because again, those steroids are fat soluble or lipophilic, but not water soluble. And so in order to transport in the blood, because the blood is primarily made of water, you're going to need carrier proteins to move that steroid hormone around. But because steroid hormones are lipophilic, notice they can easily pass through the endothelial cells of the blood vessel, and then they can easily pass through the plasma membrane of a target cell. And so those steroids then can get into the target cells and then bind to receptors. So we use cholesterol, we use enzymes found within steroid producing cells so that we can produce the steroid hormones, and then we're going to dump them into the bloodstream and then use carrier proteins to circulate them around. So steroids are not water soluble. They use carrier proteins in the blood to move around. They are going to bind to sometimes specific carrier proteins. Other times they'll bind to just something non-specific like an albumin, which is common in the blood, and that will be its carrier to get it to its target cell. Because steroids utilize carrier proteins, it's actually going to protect them from enzymatic degradation. That actually means that steroids will have longer half-lives. So if we were to analyze steroids versus just protein-based hormones, steroids are going to have longer half-lives than your protein-based ones. That means that they last longer within the blood. And again, that's because those carrier proteins are actually protecting against enzymatic degradation. So you might see something like cortisol that has a 60 to 90 minute half-life, whereas something like insulin will have a much shorter half-life in the blood. Once the steroids reach their target cells, they're going to unbind from their carrier protein, and then they're going to diffuse across the plasma membranes into their target cells. The cellular mechanism of action for steroid hormones is really based on the study of receptors that are found within the cytoplasm or within the nucleus of the target cell. That doesn't mean that steroids can't bind to other receptors. You might notice on this diagram that the steroid can bind to a receptor that's in the plasma membrane. We'll come back to that idea. But most steroids are going to enter into the target cells, and they're going to bind to receptors that are either found within the cytoplasm or they're going to bind to receptors that are found within the nucleus. And so this is going to be the beginning of our cellular mechanism of action. It's getting the steroid into the target cell and then binding to receptors that are either in the cytoplasm or in the nucleus. Once the steroid has interacted with a receptor, it actually becomes what's known as a transcriptional factor. So if we look at this portion of the diagram, here we have the steroid, I'll mark it with S, steroid, and then we have its receptor. And what it's doing is it's actually interacting with the DNA. So if we had DNA, if we draw it like this, and you have this steroid with its receptor interacting with the DNA, so this is DNA, then what you're looking at here is you're actually looking at a transcriptional factor. I'll just mark it as TF. And so a transcriptional factor is something that's going to interact with the DNA and begin the process of producing messenger RNA from a gene. So transcription is really going from DNA to RNA. And so steroids are very powerful in the sense that they can interact with DNA, produce RNA, and then if we can then go through translation, we can produce proteins. So steroids have the ability to allow cells to produce proteins. So we go through the process of transcription. I'll just put trans here, and then translation. So transcription and translation are the processes of DNA to RNA, transcription, and then RNA to proteins, translation. So steroids with their receptors are actually acting as transcriptional factors. And this really is, if you think about it, the secret of life. This is how we can activate our genes in order to produce proteins. We need transcriptional factors. And it turns out that steroids are an example of transcriptional factors. We refer to this whole process as you go through the process of binding to the DNA, producing messenger RNA, having that messenger RNA go out and be translated by the ribosomes and producing new proteins. We refer to this whole process as the genomic effect. And so activating genes is this genomic effect of cells. So being able to produce new proteins and then having those proteins go on to do work. If a steroid was to bind to a receptor that's already in the plasma membrane, we would refer to that as a non-genomic effect. And although non-genomic effects occur, we want to focus mostly on the genomic effects of steroids because that is predominantly how they exert their effects, and we know the most about those particular mechanisms. So let's look at this idea of steroids activating receptors within cells and going through this genomic mechanism. So let's talk about the idea of a genomic steroid mechanism of action. So we know that steroids are lipophilic. We know that they're going to enter into cells. So if we draw a cell, we know that the steroids are going to leave the blood and they're going to enter into the cell because they're lipophilic. And they're going to interact with receptors that are found within the cytoplasm or within the nucleus. These receptors have two parts to them. So if this is a receptor protein, if we're looking at this structure right here, if this is a receptor protein for a steroid hormone, so here's our steroid hormone in blue, there are two parts to it. There's a ligand binding domain. That's the part that's going to interact with the steroid. And then there's the DNA binding domain. The DNA binding domain is the portion that's going to interact with the DNA. So think of it as like a docking structure, like a landing gear. So we have the ligand binding portion, and then we have the DNA binding portion. Once the hormone interacts with the receptor, it's capable of then interacting with the DNA. So if this is our gene, if you will, our target gene that we want to transcribe, there needs to be a section in front of the gene where the transcriptional factors are going to interact. So this is the hormone response element, also referred to as the promoter region. So promoter regions are in front of the target genes. So when a steroid enters into a cell, it's going to interact with a receptor. And so now that structure, that steroid hormone with the receptor, is going to bind to the DNA as a transcriptional factor. And so we're going to interact with the DNA as a transcriptional factor. And the way it's able to do that is by binding to this hormone response element or promoter region. The promoter region or hormone response element is going to have two half sites to it. So there's 1 and 2 half sites. So it actually takes 2 receptors that have been activated by hormone to interact with the hormone response element. So this bottom portion down here, this is the hormone response element, and it takes the first hormone and receptor. So we have hormone, and then we have this receptor portion here. So I'll just mark it R as receptor. That's going to bind to 1 half site. And then we have another hormone, part 1 here. We'll call this the second one though. Second hormone with its receptor binding to the other half site. And so that's what's going to activate the hormone response element and then allow us to go through the process of transcription. We're going to read the gene, and we're going to be able to then produce messenger RNA. And that's a whole nother physiological mechanism. When you are reading the gene, you actually have to bring in an enzyme. So there's an enzyme that comes in and is going to read the DNA, and that enzyme is known as RNA polymerase. And it actually turns out there's multiple versions of RNA polymerase. You can talk about polymerase 1, polymerase 2, polymerase 3, and that's going to allow us to produce different types of RNA. So it turns out that RNA polymerase version 2 is the one that allows us to read target genes and produce messenger RNA. But before that enzyme can come in and bind, we need to activate the hormone response element. We need to activate this promoter region, and we're doing this through a steroid hormone mechanism of action. So let's look more closely at the full process at work. So let's look at the mechanism of steroid hormone action within the target cell. So this is the genomic effect. Again, not the non-genomic effect. This is the genomic effect, because we're going to take a hormone and we're going to have it pass through the plasma membrane and interact with the receptors that are inside the target cell. So let's go through sort of a step-by-step process here. Number one, the steroid hormone has to travel through the bloodstream. So here we have the steroid hormone traveling within the blood. This is the blood. And it's attached to its carrier protein. Because steroid hormones are not water soluble, they use carrier proteins to move through the blood. Once they get to the target cell, they need to unbind. So step number 2 here, we need to unbind from the carrier protein so that then the steroid hormone can pass through the plasma membrane and enter into the target cell. Number 3, the steroid hormone is going to interact with a receptor. Sometimes the receptors are in the cytoplasm, and again, sometimes the receptors are going to be found within the nucleus. So it depends. Sometimes you have cytoplasmic receptors, other times you'll have nuclear receptors. The point is that the receptors are within the target cell. So here we see the hormone interacting with its receptor, and now they are bound to each other. So once the hormone and the receptor have interacted together, you have formed a transcriptional factor. So again, we'll label it TF as transcriptional factor. That transcriptional factor is going to enter into the nucleus and it's going to bind to the DNA. So we could call this step number 4, where the hormone and the receptor move into the nucleus and they interact with the DNA. Of course, the DNA binding portion, so this docking portion, and the docking portion is going to interact with the DNA. That's the part that's actually going to bind to the hormone response element. So you have a section on the DNA, which is the promoter. It is the hormone response element. And then following that will be your target gene. So you have to interact with the promoter first, the hormone response element, before RNA polymerase can come in and read the target gene. In order to activate, though, the hormone response element, it takes 2 of the same steroids and receptors interacting with the hormone response element. This is known as homodimerization. So I'm going to show you a more complicated version of this. So in this diagram, it's still the same concept. Okay, we're still talking about genomic mechanism. Here we have steroid hormone estrogen entering through the plasma membrane, interacting with its receptor. But notice that it takes 2 estrogens, 1, 2, and 2 receptors, 1, 2, coming together as what's called a homodimer. So this is homodimerization, 2 of the same steroids, 2 of the same receptors interacting together so that they can bind to the DNA. So down here in this diagram, we see our strand of DNA, and you'll notice that we have one estrogen and a second estrogen and the two receptors, 1, 2, binding to the DNA. So this is that homodimer transcriptional factor. This is our transcriptional factor that's going to interact with the DNA and activate various target genes. What you'll notice, though, on this diagram is it's listing all kinds of different proteins that can be produced from various genes, because estrogen doesn't just affect one type of gene, it can affect lots of different types of genes and have varying types of responses based on the proteins that are produced. So the genomic mechanism allows us to produce RNA, and then it allows us to produce proteins. And so here we have transcription taking place to produce RNA, and then we have translation taking place so that we can produce proteins. And then those proteins can go on to give us the steroid hormone response. Because steroids can affect lots of different target genes, you can get varied types of responses within the body from the same steroid. So estrogens have many effects within the human body. Testosterone has many effects within the human body. Cortisol can have many effects within the human body because they can affect multiple types of genes. Now, this is an oversimplification of the process. There certainly are many coactivators and co-repressors that can affect the transcription and translational process. For example, with estrogens, there's lots of selective estrogen modulators, but this gives us a general idea of the steroid hormone activation mechanism. But again, when you look at a more complex diagram, you'll notice that there's, again, lots of different proteins that can be affected by the effects of estrogen. So it turns out that in medicine, we can use various types of drugs to affect estrogen receptors and try to get the appropriate responses that we're looking for. So, for example, you can look at drugs like tamoxifen, raloxifene, which are actually selective estrogen receptor modulators. And these drugs can affect the body in different ways depending upon what tissues the drugs are interacting with. For example, tamoxifen actually has anti or negative estrogen effects in breast tissue, but it actually promotes estrogen effects, or it has a positive effect on bone tissue and endometrial tissue. Raloxifene, another estrogen medication, actually has positive estrogen effects in bone tissue, but it doesn't affect endometrial tissue. So depending upon what types of effects you're looking for, you can treat conditions like breast cancer, osteoporosis. You can even get into treating heart disease based on the selective estrogen modulator medication that you're using and how it has positive or negative effects in specific body tissues. So that is the power of some of these medications, but it really comes back to the power of estrogens and testosterone and progesterone and cortisol effects within the human body. Because these have genomic type effects, because they affect genes, they can affect lots of different mechanisms physiologically in the body. Now, steroid hormones are not the only hormones that can utilize these types of effects where you can act as transcriptional factors and affect the DNA. It turns out that there are amine-based hormones that can work this way, specifically thyroid hormone. So what are amine-based hormones? Amine-based hormones just simply means hormones that are derived from single amino acids. Some examples of this would be melatonin. Melatonin is derived from tryptophan. It's produced in the pineal gland. You have epinephrine, norepinephrine, which are catecholamines, which are derived from tyrosine. And then, of course, you also have the thyroid hormones, which are also derived from tyrosine. So on this particular diagram, this is the amino acid tyrosine, and we can see that tyrosine can produce the catecholamines. This would be chemicals like dopamine, norepinephrine, epinephrine, amine-based structures. And then we can also produce thyroid hormones. I'll have a whole nother video on thyroid hormone synthesis and how you attach the iodines and produce T3 and T4 thyroid hormone. But these are amine-based hormones. And it turns out that thyroid hormone works very similarly to the way your steroid hormones work. They are lipophilic, so they pass through the plasma membrane, and they're going to interact with receptors that are found within the target cell. So let's take a look at thyroid hormone as a mechanism of action. So thyroid hormone is actually produced by the thyroid gland, and then the thyroid gland is going to dump your T3 and T4 hormones into the blood. And then they're going to utilize a carrier protein. It's actually called thyroid binding globulin. In this example, it's showing us the T4 attached to the thyroid binding globulin. And that's going to make its way to the target cells, and then it's going to release from the carrier protein, and it's going to pass through the plasma membrane and enter into the target cell. So you could sort of refer to this as sort of steps 1 and 2 if you were looking at this as a step-by-step process. So we produce T3 and T4, they're dumped into the bloodstream, they're going to bind to a carrier protein, and then those carrier proteins are going to take them out to the target cell because they are not water soluble. So not water soluble. So again, very similar to steroid hormones. Once you get to the target cell, you're going to detach from the carrier proteins, and you're going to pass into the target cell. Step 3, you are going to have your T3 hormones interact with receptors, or the T4 is going to be enzymatically converted into T3 because T3 is the active version of thyroid hormone. So T3 is your active form of thyroid hormone. So there's an enzyme. It's a deiodinase enzyme that's going to take off an iodine and turn T4 into T3, and then any T3 that's already in the cell can find its way to its receptors. So in this diagram, it's actually showing you another carrier type protein, this binding protein, that's going to help move the T3 through the cytoplasm. We're actually going to see this translocation into the nucleus. And then this is where you find the receptor. So this is actually a nuclear hormone receptor that's going to interact with your T3. So here's our T3 hormone, and it's interacting with its receptor. And so now you have your transcriptional factor. Once you have the receptor and the hormone bound to each other, you have a transcriptional factor. So we could say something like step 4, the T3 uses a carrier protein within the cells to move into the nucleus, and then it's going to find its nuclear hormone receptor. Once you have a transcriptional factor, you need to interact with the DNA at the hormone response element. So what we have on this diagram is we're zooming in on the DNA. Remember that the hormone response element is also the promoter. So this is the promoter. And the promoter is going to be interacting with two receptors on each half site. Now, thyroid hormone is a little bit different than what you see with steroid hormones. Steroid hormones use a homodimer situation where two of the same steroids, two of the same receptors interact with the hormone response element. It turns out with thyroid hormone, you use thyroid hormone, so T3, interacting with the receptor. This is the first transcriptional factor. The second transcriptional factor, though, is something that's different. This is actually a vitamin A derivative. Vitamin A derivative. This is known as 9-cis-retinoic acid. So it's actually produced from vitamin A. And it's going to interact with its receptor. So here's the 9-cis-retinoic acid that's going to interact with its receptor. And that's going to form the second transcriptional factor. So because both of these are different, this is known as a heterodimer. Hetero meaning things that are different. So homo, same, hetero, different. So here we have the first transcriptional factor and the second transcriptional factor, two things that are different from each other interacting with the hormone response element. Once those two different structures bind to the hormone response element, then you can bring in your RNA polymerase, and then you can have transcription take place. So transcription then will lead to the production of messenger RNA. So think of heterodimer as like step 5. You form a heterodimer on the hormone response element, and that's going to allow you to produce messenger RNA. And then messenger RNA is just like in steroid hormones. It's going to leave the nucleus. It's going to go interact with ribosomes. You're going to go through the translation process, and you're going to produce the proteins that are going to give your thyroid hormone response. So if we go back to this simplified version here, this shows us the first transcriptional factor. This is T3 interacting with its receptor. What it's missing is it's missing the vitamin A derivative, the 9-cis-retinoic acid, interacting with its receptor as it binds to the hormone response element. So that would be the heterodimer that then is going to allow for the transcription of the target gene. So once you have transcription, you produce the messenger RNA. Messenger RNA leaves the nucleus. Then you have the translation process where you produce your proteins. And then those proteins can give us the thyroid hormone response. So vitamin A is actually being utilized along with thyroid hormone in order to produce the proteins that we need in the target cell to give us the thyroid hormone response. You'll also find that other vitamins do these similar processes. So vitamin D will actually work in other heterodimer responses so that you can produce messenger RNA and then produce proteins and get other cellular responses. So that was steroid hormone mechanisms of action and thyroid hormone mechanisms of action through what's known as the genomic mechanisms. So I hope that helps. Take care.